Bom dia a todos. It's a great pleasure to introduce Rodolfo Gómez. Rodolfo is the head of a lab in Santiago de Compostela devoted to the study of musculoskeletal diseases. Rodolfo has done his PhD in Santiago and in Newcastle, and then he's a group leader for more than 10 years. And his lab studies mainly osteoarthritis, rheumatoid arthritis, and also rare metabolic bone diseases. Rodolfo has hundreds of publications, lots of patents. He's really willing to transfer his knowledge from what is basic science to really clinical approach and more technological approach. But with all these things that Rodolfo has accomplished, and he has a very well-funded lab and a large one, he also has two different things that I would like to highlight. One is that with all these years of science, he's still very curious and he's still very passionate about doing research, as you are going to see. And the other one is that his lab is like a family. They are all very connected. They are all very close to each other. And this is a very fruitful environment to get the success that Rodolfo has. And it really makes a difference. So we work together at Target. It has been an immense pleasure to collaborate with Rodolfo. I think it's the beginning of a long-term collaboration. We are very, very grateful, Rodolfo, that you came here and did you accept our invitation. And Rodolfo is going to focus on his long-term research on innate immunity, on TLR4, on endotoxemia, that connection with what happens in our joints. So thank you, Rodolfo. It's a pleasure.
I don't need it. You hear me? Yeah. Well, thank you for the introduction. For me, it's a pleasure to be here. And coming to Lisbon, to Portugal from Galicia, is like being at home. I think Saramago was right. And well, today I'm going to talk about targeting innate immunity and bone metabolism in rheumatic diseases and what we can bring from bench to the clinic. And specifically, I'm going to talk about how I start researching in innate immunity and what I learned during this path. Well, this is me. This is my institution. First, we need to talk about the size of the problem that we have in our hands. More or less, rheumatic diseases are a cluster of 250 different diseases. And among these, we have osteoarthritis, rheumatoid arthritis, gout, systemic lupus. And it is considered that these pathologies are among the most prevalent diseases worldwide. I know that you know better than me what we are talking about, but just osteoarthritis is the major cause of disability in developed countries. Therefore, they are tough diseases. But one of the scariest parts of these diseases is how they impact in the quality of life of the patients. In Spain, there is a study called EPICER. They compare different chronic diseases and how these diseases impact in the quality of life of the patients. And among the worst diseases were the rheumatic diseases. As you can see in the upper graph, they impact in mental health and in the physical health. Therefore, if we combine this data with the data of the prevalence, we have in our hands a really, really major problem for the healthcare system and for the society. And I asked the Spanish Society of Rheumatology for a few relevant pictures that put some information. You know, image sometimes is better than words. And despite the different etiology of these different diseases, they share some molecular patterns between them. And we are talking about specific features of the inflammation and also specific metabolic changes in the bone. And the bone is something relevant for the rheumatic diseases because it's associated with fragility and bad quality of life. But it has been stated that maybe 13 risk factors support this common metabolic alteration or these common similarities. We know that aging is one of the major factors, certain genetic profiles, unhealthy habits, but one of the risk factors that always pop out is the metabolic syndrome. It's a cluster of diseases that that behave as like comorbidities of the rheumatic diseases and strongly impact in the health of the patients and in the progression of this disease. But one of the interesting things of metabolic syndrome is that it develops a vicious cycle with the rheumatic diseases in terms that rheumatic diseases are associated with more sedentarism. These, together with unhealthy diet, altered microbiota, are promoting this vicious cycle and the contribution to the development of metabolic syndrome, which is composed by obesity, dyslipemia, hyperglycemia, and hypertension. But 20 years ago, we didn't know how to connect the incidence of metabolic syndrome and the rheumatic diseases because there were a lack of a lot of research. In that time, when I was young and I have hair, you will see, we were talking about, we would start working with obesity. And 30 years ago, obesity, fat tissue, it was almost just a deposit tissue for energy, for lipids, or for energy, or an isolating tissue, like, you know, protecting you against the bad weather, like this one. And but in 1994, it was discovered leptin, and it was totally changed our point of view. Because the fat tissue is a true endocrine organ. Therefore, this tissue can produce cytokines, chemokines, but also a massive amount of different types of adipokines, which are adipose tissue-derived products. And these molecules are able to control function all across the body. Therefore, imagine if you have a pancreas 8 times bigger than it should be, or a thyroid bigger than it should be. Therefore, this is happening with obesity, which is one of the core members of the metabolic syndrome. But at that time, 20 years ago, nobody tested the connection of this endocrine function of the adipose tissue with rheumatic diseases. And we said, okay, we are going to have a look. And it was one of my first papers. At that time, I have hair, a lot of hair. 20 years later, I am like this. This was the first time that somebody described a link between adipokines and rheumatic diseases. And I was really young, and I was so happy with these results because opened new frontiers, new possibilities. We determined that rheumatoid arthritis patients has more elevated adipokines levels in the blood. It was life-changing, and we started to say, okay, they have more. What these adipokines do in the blood of the patients and in the joints of the patients. Okay, the next year, after this paper, we start to treat chondrocyte cells with leptin. And leptin, it seems that alone, it do not anything. But when you combine with typical inflammatory factors in the joint, it's like gasoline and fire. Boom. You have 30, 40%, or even 200% more production of innate immune responses. Here, the graph is with interferon, but we tested with interleukins, with all the different factors. It was amazing, but we didn't stop there. We jumped from here to adiponectin, which is another adipokine. And we identified that in the chondrocytes and other cells of the joint, there are receptors that sense these adipokines and start to react against this activation. Interestingly, one of when we stimulate these cells with these adipokines, always innate immunity go up. Happened with leptin, happened also with adiponectin, and say, okay, there is something, this is like a pattern in this. We still were working with more papers, and I just picked a few of the cool ones. And then in 2011, we also studied how these adipokines were able to recruit more immune cells, because at the end, we don't want too much immune cells in the joint. And they produce interleukin-8 as water. This was really interesting. But in 10 years after the first paper, we arrive here and we publish just our work and the work of other peers. And nowadays we have tons of information pointing that adipokines, here is the leptin, but we have the same for other adipokines, are master regulators of the immune system. They do a lot of things in adaptive immunity, which is relevant for the autoimmune diseases, but also they elevate the responses of innate immunity, which is something and a specific that is going to affect all the diseases. Because this, we have a different branch of research in the innate immunity, and it is something that maybe it's underlying, maybe it's the underlying mechanism that is driving a few of the phenotypes of these diseases. It's not the only one. You know, we are talking about complex diseases, and this is not just, this is a gene that I turn on, turn off. It's more complex. But innate immunity is like a mechanism of defense that is is highly conserved. It's rapid, fast, and nonspecific. It's like Marines. I hit first, and then I ask. Therefore, with this repertoire of receptors, we can sense microbial-derived products, but also destruction products that came from the host due to destruction of the tissues. We have this type of pattern recognition receptors, toll-like receptors, node receptors, and also inflammasomes. And it seems that they are in the core of a lot of rheumatic diseases, the innate immune response. But among these receptors, this is my favorite, this is my jam, is TLR4. And TL4 is like a very promiscuous receptor. It doesn't matter. You know, it can detect virus, bacteria, wherever. Chunks of proteins unfolded. It's like a specific way to detect that something is, that there is a problem. It's like this sensor that we have here, that when there is smoke, it doesn't matter the smoke comes from, it always, okay. With this in mind, once it's activated, this receptor, it elicits a massive response, Just a simple idea. When we take cells in the lab and we stimulate TLR4, and you give the cells indomethacin, high doses of indomethacin, you cannot control the inflammation. It's the same. It's like water. It's, you know, it's too big to represent why when you provide a few nights to patients, you don't see any change if you have this on, because never go down. Even with corticoids, it's difficult to control this. I tested in the lab. It's black and white. Well, this receptor, as I said before, is like a master regulator of inflammation. And it can sense DAMPs, which are damage-associated molecular patterns, which are products released upon the destruction of the tissue. Okay, we have a tissue that is being degraded, and chunks of the proteins are being released, and these chunks of protein are going to go to the receptor and activate the receptor. In this list, I have, it's a little bit old, in 2015 when I did it, but we have chunks of fibronectin, fibrinogen, oxidized LDL, which is a link with metabolic syndrome. If you have oxidized LDL, you are going to have more activation of the TLR4, which is also interesting with the TASTY trial. But also, more recently, in the last maybe 10 years, 6 years, there is a lot of work about the activity of the endotoxemia, because this receptor can also detect pathogen-associated molecular patterns, which means that it's not like I am going to detect this specific part of the protein. No, it's like I detect something related with this. It's like a diffuse, and it works. And it's having described that gut dysbiosis, alter intestinal permeability, and these microbial-derived products jump to the blood, go to the joint, and help to progress the disease. And one of our colleagues, one of these guys, the second one is from Oporto, determined that the levels of LPS in the blood are associated with the severity of the disease. which is quite nice. Again, always we are talking about that this always is popping out in rheumatic diseases. Well, I did I picked a few papers that can be useful in order to depict what happened in 20 years, more or less. There are key papers that point that the innate immune responses are key for these rheumatic diseases. Among all the studies, I totally in love with the work of Abdullahi, difficult to pronounce, because they did an animal model, and an arthritis model driven by interleukin-1 expression. And when they shoot off TLR4, suddenly the severity of the disease go down. It was amazing. It was so clean, so pretty. And also, Scancelo, in osteoarthritis, she determined that when permeability of the synovial tissue is altered, plasma proteins cross from the blood and to the synovial fluid and start to activate TLR4 all across the joint, which was something pretty nice, because this activation was all across the tissue, in bone, in synovium, in tendons. And one of the interesting things, as I mentioned before, is that, as you can see here in red, that always is alteration in the bone. You have more bone erosion, subchondral bone alteration in osteoarthritis. This is something that needs to be studied. And we did this during the last years. Different colleagues and our group, we were testing in what TLR4 activation, where it takes place within the alteration of the rheumatic disease. And we see that when you stimulate mesenchymal stem cells, in the bone there are two main equilibriums. The cells must be choose between become a fat cell, adipocyte, or an osteoblast. And also there is an equilibrium between the cells that destroy bone, the osteoblast, and the osteoclast. Therefore, we have two equilibrium. But what activation of TLR4 do is, I'm going to take this mesenchymal stem cell, instead of become bony cell, I'm going to put this cell in a adipocyte cell fate. And additionally, I'm going to promote osteoclast. Therefore, at the end, I have a patient that due to the activation of TLR4 during maybe 15, 20 years, their bone are going to be like mayonnaise, full of fat and really, really weak. You can do click. And additionally, as I mentioned before, metabolic syndrome is something really complex. A lot of different diseases. We start with obesity, but there are also other phenotypes in the metabolic syndrome, like the affectation of the liver. And we start thinking, okay, maybe if it works with leptin, maybe we should try with other parts of the metabolic syndrome. And we arrive here with an hepatokine, which is mainly produced by the liver, and it has been described that it's elevated in obesity, but in fat liver, in diabetes, and hypertension. And you say, what? It's really interesting. We went to the synovial fluid of the patients, and it seems that in the synovial fluid of the patients, it's increased in the osteoarthritis patients, in the gout patients, and in the pseudo-gout patients. And it correlates perfectly fine with hypertension and diabetes. Okay, we say, okay, this is really interesting. We go to the lab, and we put this RBP4 on top of cartilage cells, and also on top of synoviocytes. And it seems that it behaves as a dump. It activates TLR4. And as I mentioned before, TLR4, once you activate TLR4, you have a problem. And then we study more cells, and in the synovial tissue, it activates inflammatory markers, but also something really, really powerful. It is a massive generator of neutrophic chemotaxis. And in the cartilage, it's switching off mitochondrial environment. Therefore, it's like asphyxia. I don't know how to say if I am saying properly, but it's a lot of inflammation for the cartilage, and the cartilage it's breaking down. But this is one of the, we have been studying how the adipokines, how the hepatokines impact the innate immunity. But this was almost 20 years ago also. I also have this here. And when we tested in the opposite way. Could the innate immunity impact certain hormones that are important for the metabolic syndrome in order to, to close the vicious circle? And it seems that it seems an easy experiment, but the consequence of this was really big. For example, we stimulate the cells, joint cells, with LPS to activate TLR4. And then we study an enzyme is called GOAT, which is the enzyme that put a seal change in ghrelin. Ghrelin is the hormone associated with the appetite, and it's a secretagogue of growth hormone. And without this activity, the ghrelin go in terms of activity, which means that we have innate immune activation, we have low levels of GOAT, means that ghrelin is not active, and if we don't have active ghrelin, we have low GH signaling, we have low appetite, and the reduced anabolic effect of this hormone, which somehow can be related with inflammatory cachexia, because at the end, if you are pushing, you have quite a lot of activation of innate immunity, you have low levels of GOAT, you have less activated ghrelin, you're going to have these potential problems. This is just the summary, more or less, in this chunk of this information. It's like innate immunity acts as a convergence hub linking metabolic syndrome and endotoxemia to chronic rheumatic diseases. But with the existence of maybe more than one, a loop between when you have destroyed the tissue, this is generating dumps that activate more innate immune system, and then this go back again and again and again. Therefore, we need to cut somewhere this change, because otherwise we are not going to, or you change the metabolic syndrome, or you control the gut dysbiosis, or you control the innate immune. But we need to control something, because otherwise, if you have this on for maybe 15 years, it's not 1 year, it's not one time. It's like every single day. Okay, you would say, okay, Rodolfo, we need to treat something. We need to do something. We have something, but petit. We have something we have interleukin-1 blockers that can help somehow, but it's downstairs of the signaling. We can control somehow the colchicine, but take care with colchicine, because in the lab, when you see what the colchicine do to the cells, it's scary. And we have hydroxychloroquine. We know this friend in the COVID, but we miss TLR4 inhibitors, we miss MD2 inhibitors, CD14 inhibitors. I know that this is scary to touch the innate immunity, because it's something really is like the basement of the building of the immune system in unspecified. But we need to do something with this. Okay, and we start to think, okay, what we should do? Because at the end, we just have a lab. And we start to think, well, maybe we could do some drug repurposing, because we are developing new drugs, but it takes more than 20 years to arrive from the bench to the initial steps of the clinic. We are in the middle of this, and sometimes it's so frustrating. Therefore, we say, okay, maybe what happens if we could use what we have in the clinic? In the meanwhile, we develop new treatments. And using computational pharmacology, we recreate the TLR4. It's an heterodimer, it's like two little umbrellas like this. And we start to make the dockings of the LPS to this receptor. And we identify where it binds, where is the hole of the key. And then we confront this information with a database with a lot of drugs. And something pop out and say, oh, fantastic, when I received the news. It seems that amitriptyline, which is antidepressant, well known, pretty old, more or less the safety is quite nice. And we check that it binds to TLR4 even better than the LPS. Say, okay, this is something. I simplify this because this is madness. What we see here, then we decide, okay, we are going to take cells from the patients, we are going to stimulate with LPS in order to activate the innate immune responses that cannot be controlled with corticoids or with indomethacin. And we are going to put amitriptyline. And all the receptors, all the proteins, all the genes were downregulated with the amitriptyline, which means that amitriptyline was stopping the innate immune response, the gene changes due to the LPS stimulation. And then we say, okay, yes, but you cannot say that you have something in your hands just with a couple of genes. You need to do something cool and big. And therefore, we test all the proteins in the cells that were changed due to the stimulation of LPS in the presence or absence of amitriptyline. And then we go to the bioinformatician, Andres, and we start to figure out what these proteins tell us. And it seems that it truly shut down the inflammation mediated by TLR4. And among all the pathways that it interfere, we identify that obviously activated TLR4 signaling is downregulated, but also interleukin-1, which was, I know that they share some signaling pathway, but it was interesting because in the context of, we have, it's a known target for rheumatic diseases, but also the inflammasome. We say, okay, this is pretty nice results. Maybe amitriptyline is shutting off interleukin-1 signaling because it binds to the receptor. We did it again, but it didn't work. The amitriptyline do not bind to the interleukin-1 receptor. I say, okay, it's switching off into interleukin-1 signaling and it's not interacting with the receptor. Maybe it's the activity. Maybe it's TLR4 inhibition, what is associated with this. And we use a compound, the Cli95, which is a tool in the lab, a pharmacological tool to block TLR4. It was tested in the clinic, this drug. It was discontinued. It was in phase 3, but at the end, it didn't work properly for sepsis. But we tested, and it works. It means that if you turn off TLR4, you turn off also interleukin-1 signaling, which is quite nice. We have only one target and 2 effects. But obviously, we didn't believe in just a few genes, and we did again the same scenario. We take cells, we stimulate, instead of with TLR4, we stimulate cells with interleukin-1 beta, and we co-stimulate with those different doses of amitriptyline. And we have, again, a dose-response inhibition of the effect. We go to the protein, we did the same, pathway enrichment analysis, and again, TLR4 signaling downregulated, interleukin-1 signaling downregulated, obviously, because we have just seen. But again, inflammasome downregulation. And we say, okay, it's interesting. Two different stimuli, same common response due to the amitriptyline. Maybe inflammasome is taking place. It's doing something nice. And we study the expression of the inflammasome. And we use different tools, small interference RNA. We use Cli again to block the receptor. And it seems that when you block TLR4, the proteins of the inflammasome are downregulated, are less produced. There are lower amount of this inflammasome. And because that, when you turn off the inflammasome, you can block at the same time TLR4 signaling and interleukin-1 signaling. This is quite nice. But provide us more information. It seems that TLR4 activation tone is modulating the levels of the inflammasome. You have more TLR4 activation due to maybe endotoxemia, due to maybe you have a degrading tissue, maybe because you are fatty and you have a lot of adipokines. Maybe you are going to have the tone level of the inflammasome like this. And this is like gasoline when you have an autoimmune disease or something, it's just like burning. Everything is prepared for this. And it seems that it was the mechanism underlying. But yet, again, I am a health researcher in a hospital. I am part of the health system in Spain. Therefore, we must always ask this and this question: what is the clinical relevance? Okay, you play with your toys in the lab, you do fancy pictures, you go to Congress, but yeah, at the end of the day, what can be transferred to the clinic? Okay, we start thinking, okay, maybe since we have quite a lot of information with the proteome analysis, we take all this information and we put it in databases to compare the proteomic changes in the cells with the proteomic changes that have been described for different diseases. And interestingly, the list of the diseases that pop out are arthritis, gout arthritis, osteoporosis, and osteoarthritis. This is fantastic. It fits perfectly with my presentation in Lisbon, maybe 10 years later. But you know better than me that doing science in the hospital is really tough because you need to have a lot of permits, bureaucracy, time, and do data mining is really complex. We think maybe we could look for osteoarthritis correlation with amitriptyline. It's difficult because osteoarthritis is like a heterogeneous disease that coming from is really difficult. We have at least 5 phenotypes, difficult. But using data mining with gout is maybe it's more simple. And since we didn't have access to the clinical records, I have access to the clinical records, but only one by one patient. I can access the patient of my hospital and check what is happening there, but only one by one. I need raw data. I need massive data. And I went to the department of the hospital and I say, okay, can we have that data with no name of the patient, with just the age, the sex, and the consumption of colchicine, because colchicine is mainly used for gout. It's not only, but it's mainly used for gout crisis, and also amitriptyline. You say, okay, if you just need the purchasing of this, who is taking this in the clinic, I can give you, because it's not, it's aggregated data. It's just this number takes during 5 years this thing, and we did this. And we identified this. We have a cohort of 107,000 patients, men and women above 65 years. We have the 450,000, but we need to cut it because otherwise we are going to take the colchicine that it was used for other uses. Above 65 years is more gouty use. And we test if there is a correlation or a relationship between the use of colchicine and amitriptyline. And it seems that the patient that takes amitriptyline are more prone to use colchicine, which is quite nice because at the end, it seems that if you are taking amitriptyline, you take less colchicine. And why you are taking less colchicine? Maybe because you have low inflammation levels, as I mentioned, as I showed you before, at cellular level. And then we say, okay, we are going to, as you can see here, we don't know, we have amitriptyline draft boxes because it's the data that we access, how the patient take boxes. And this is from 15 years, 5 years of consumption. There is a huge data here. And it seems that there is a negative correlation. It is pretty nice correlation. When you start taking amitriptyline, as much as you take less colchicine. And it was quite nice because at the end, we present this in the Spanish Society of Rheumatology, and they were quite happy because we received, when you need to prescribe an antidepressant, because it's a big comorbidity of rheumatic diseases, you can say, okay, maybe I provide you this amitriptyline treatment as an antidepressant, and maybe I am killing 2 birds with one shot. But we didn't stop, 20 years is a lot of time, and we didn't stop there. We tested, we did the same with other drugs that popping out in the list. We did it with naloxone and thalidomide. I know that thalidomide is a tough drug, but it's interesting to know that it can do this. And And then we also have this we talk about innate immune regulation, but also the phenotype of the bone is interesting to see what is happening. You know, in different diseases we have some problems with the bone. We have low bone density, we have bony adiposity, which is associated with fragility. Therefore, it has been described that gout patients, despite they have bone erosions, they have also bone growth in certain parts. And it was quite interesting. Somehow it has been related with the use of colchicine. And we say, okay, this is interesting, but nobody tested if colchicine is able to control the equilibrium between osteoblasts and adipocytes. Because at the end, when you have the patient in the surgery room and the bone is full of mayonnaise, this is the main problem. You need to block this. Otherwise, this fat is going to block the ability of the bone to recover, and it's going to produce a lot of adipokines. And we tested in the lab, and we identified that colchicine strongly block the adipogenesis in the bone context, which is really interesting. But, and also we published this, but about this, it's necessary to be aware that colchicine is a tough guy. It's more dangerous than it seems, because at the cellular level, it's doing awful things. If you have intoxicated with colchicine, it's not easy. You are going to have a bad time. Again, you're taking the advantage that we have other line of research of therapeutic ossification. We, you know, in spondyloarthritis, even we have syndesmophyte, osteophytes. Also, we have in gout, we have abnormal bone growth. There is a lot of information about this. And spasticity, it was in the past associated with therapeutic ossification and the drugs to treat the spasticity. And it seems that among all the drugs, baclofen is promoting osteochondrogenesis in the mesenchymal stem cells. And this is quite interesting because it has been, it was recently published this year, yeah? And it provides information for the clinic. And it downregulates adipogenesis, even the adipogenesis induced by the TLR4, which is quite nice. And okay, and you can say, okay, we are talking about when you have low amount of bone and you want more bone. But what happened in the other way? What happened when you have abnormal bone growth, and you don't want it? Okay, and here this was more complex, and we need to combine 3 different drugs: oral antidiabetic, corticoid, and indomethacin. And the combination of these 3 drugs, we also patented, is able to block this commitment of the cells toward the bone phenotype. I know that for sure that it has been tested in the clinic, and it seems that works, because at the end, since they are repurposed drugs, you can test it, despite this patent. But we didn't stop there. You know, we were quite prolific in the last 20 years. And, you know, nutraceuticals always have been associated with the management of rheumatic diseases. Extra virgin olive oil, ginger, Boswellia serrata, has been associated with improvements in the clinic. And we say, okay, maybe there are compounds there that are really, really interesting in terms of regulation of the innate immunity. And we tested. We test different polyphenols, oleocanthal, 6-sugarol, beta-boswellic acid, and we published that they can really switching off the innate immunity, which at the end is again more tools, not the coolest one that you know that are in all the news, but at the end you have more tools to play with the patients. And finally, in this section, we have been studying cell differentiation, mesenchymal stem cell commitment to osteoblasts and adipocytes, and inflammation for a long time, and we learn a lot. And during this time, in our group, there are rheumatologists, but there are also surgeons. And the surgeon said, okay, I want to play also. We say, okay, what happened if we could do better implants. You know, if you are a smoker or if you are a diabetic, your bone is kind of rubbish. Therefore, you need to do something. Or maybe if you have metabolic syndrome, obesity, or aging, or you have a rheumatic disease, at the end you are going to need an implant. And they're going to put you the same implant as a healthy, young, you know, strong man. It's not, therefore we say, okay, maybe we should, it will be decided for an implant. We would like to have control of inflammation because we are going to avoid the aseptic loosening. We like that the bone, that the cells around became bony because are tough, and you can you have more osseointegration. And then we realized that the surface of the surface are changing of the metal are changing the phenotype of the cells. If you put here a cell and the roughness is different, the cells are going to become differently. Therefore, we tested several different combinations of nanotexturization using an automatic laser on titanium surfaces. And we tested how the innate immunity and the differentiation. And at the end, we achieved nanotexturization, like a laser configuration, that once applied to a titanium surface, can reduce inflammation, more than 50% of inflammation in the cells, osteogenesis increasing, and inhibition of adipogenesis, which is quite nice, and we publish it pretty nice in this field. And well, what we are doing now? Well, I am with Patricia in EndoTarget, which is a nice project. Hopefully this year we are going to have more results, and we are looking for the impact of endotoxemia in rheumatic diseases. And something that is maybe is going to be published this year. It's one of the works of one of my postdocs, PhD student / postdoc. We are taking the cells, we are changing the mesenchymal stem cell, human cells, we are differentiated to osteoblasts and adipocytes. Because at the end we need tools to control the differentiation of the cells. If we are able to see what is the switch of this, then we can design drugs and activate or inhibit. And by using this method, we have analyzed the epigenetic changes. Then with computing systems, we are detecting the transcription factors moving this, and we identify that 6 transcription factors only working in the bone are able to control this phenotype. And it seems that when we went to the patients and we test the bone, not the blood, not the fat tissue, because you're not going to see anything outside of the bone. When you go to the bone of these patients, different diseases, we have realized that, again, the metabolic syndrome is associated with these 6 transcription factors, and these transcription factors are upregulated in the bone of patients with these problems. Therefore, when you stimulate in rheumatic diseases, this is really relevant, because at the end you are going to have 6 transcription factors that should be muted that are active. When we mute it, as you can see here, this is a staining of adipogenesis. You know, oil red is staining the lipids. These are mesenchymal stem cells that we differentiate to adipocyte cells. But when we turn off these 6 transcription factors at the same time, if you only do one, you're not going to see anything. Therefore, we have new therapeutic targets for alterations. We have from Brazilian herbs, we have 3 new compounds, really tough. And this is my jam. This is in my heart. We are working with innate immunity and hereditary ectopic ossification. We have a national project about this, and we have a close relationship with families. And it seems that we have a clinical case of progressive osseous heteroplasia. We have also cases with fibrodysplasia ossificant progressiva, but this is touching. We have 2 twins, one of with the same mutation. One of the twins have a massive bone formation in the fat tissue, the subcutaneous fat, and the other more or less is normal. And due to the surgeries, we have been able to access to this bone of this patient. And we work so hard, and we identify what is happening inside the tissue. Till the level that now we are currently testing drugs and genetic engineering to using COVID mRNA-like delivery to try to correct the phenotype of these girls. Maybe we are not going to be fast enough to avoid the death of the patient, but we have learned so much that this information can be overlaid on rheumatic patients, because at the end we know now the switching, the interruptors, the switches of this process into this disease that can be extrapolated to other abnormal bone formation in rheumatic patients. And muito obrigado. This is my team, pre-docs, postdocs, the bioinformatician, rheumatologist, traumatologist, rehabilitating physician, and these are the funding bodies. And thank you for your time. I don't know if I am out of my time. Thank you.
Thank you so much, Rodolfo. It was a fascinating talk. Questions from the audience?
Thank you. Is this on? Okay, I'm not sure if it is on, but we'll speak like that. Thank you very much. I saw that you had several papers with Juan Gómez Reino.
Yeah, it was my thesis director.
Oh, okay, because he came here several times, and we had for many years a collaboration. So
he was the director of my thesis.
Okay, so give our regards to him. There are 3 points that I would like to understand a little bit better. So the first one is the scheme that you showed with the TLR4 and the effects on the deviation of the phenotype to adipocyte on one side, instead of going to osteoblast. And on the other hand, the effects on the activity, I suppose, on the osteoblast favoring the production of rankle and inducing osteoclast. Was this what you showed? So did I make the correct interpretation? So TLR4 stimulation with the deviation to a phenotype of adipocytes, and at the same time the stimulus of on the osteoblast surface.
Is what is happening.
Yeah.
But there was a debate about the role of TLR4 in monocytes that can become macrophages or can become osteoclasts. You know, osteoclasts are the fusion of monocytes, more than 3 with this all this phenotype. But it was a few papers that say that depending in the time of the window, if the timing is really fast, if you put TLR4 activation in a monocyte, you have macrophages. But if there is a delay, a little bit of delay, for example, that you have a pre-commitment of the monocyte to become osteoclast, and you put TLR4, you have osteoclast all day, all night.
Okay, so that is a direct effect on TLR4 on the surface of the monocyte-like cell, is it?
I don't know for sure.
Or is it dependent on the surface of osteoblast? Because that's the reason I made the question. So
this here.
So this would mean that if we had a pro-inflammatory environment, we would have more adipocytes in the medulla, and at the same time, the TLR4 activation, I assume that is on the surface of the osteoblast, inducing the production of rankle. Is this the case?
I think the effect is going to be more or less when this is formed. I think if you have monocytes here and an arrow here, the effect is here.
It's a trigger on the right hand side.
Not here.
Not there.
Not here.
On the
because
right-hand
at the end you need to have monocytes to form this. And therefore, the effect of this is here. But here I wanted to show that there is an imbalance of the activity of this against this.
Understood. The other thing was the amitriptyline colchicine story. So a little bit on the devil's lawyer side, we could assume in a more clinical perspective that if we look on the right-hand side, so less colchicine in patients classified as having gout, because we are here classifying, maybe in fact they're getting more amitriptyline because in fact they have more of an amplification pain syndrome, not exactly gout. So just playing on the devil's
No, you're right, you're right. We asked the same question. In fact, the reviewers of the paper, British Journal of Pharmacology, asked the same. And we say, okay, we know that amitriptyline is somehow works as a painkiller. But at the end, when you provide colchicine to the patient, it's because you have the patient with a joint like this. They have more pain, for sure, but they have like this. And at the end, you can hide this massive inflammation. And at the end, when we made the calculation with the drug boxes, it's not, cannot, for example, when you provide colchicine, you have a lot of peaks of crisis when it's the setting down, the levels of also when you have, when you are controlling the uremic levels, you have also peaks of inflammation and they are being controlled by amitriptyline.
So this is a consumption of colchicine during episodes of gout crisis?
We don't know for sure. It's the consumption of colchicine in a window of 5 years.
So, because as you know, a lot of patients are on colchicine in a steady way, low dose, etc. So, and those patients sometimes have complex conditions where you use colchicine in a little bit like, let's try to help the patient somehow, okay? Just
right. But here you have the box, the consumption, above a certain degree of consumption of amitriptyline, you are no longer need consumption of colchicine. You can see? Above a certain level of amitriptyline, because you need, we did the calculation of what we tested in vitro, and we need to provide in the clinic, and you need a strong amount of amitriptyline to have effects. But when certain patients that need a lot of amitriptyline due to depression or whatever, it seems that above a certain amount of amitriptyline, you don't need colchicine.
I just want to add one question. You mentioned twice in your talk the terrible effects of colchicine on cells.
Yeah.
And we are very comfortable in prescribing colchicine, and from the drugs that we use colchicine for us, it's a no-brainer. So can you describe a little bit what were you
just if you put colchicine and there are tons of papers in the literature about the concentration that you can use of colchicine in the cells. You put colchicine in these doses. For this experiment, we reduced at least 20 times the concentration in the other paper. The cells died.
We are talking about concentrations that we are not using in clinical practice or not.
Depending, because when I am a pharmacist, and when you try to determine the concentration of a drug based on the levels in the tissue is difficult because we have different compartmental distribution. This is kind of complex. I am not sure if it is true, but for sure, for example, you take mesenchymal stem cells, you put colchicine in a dose, normal dose, that is potentially even below the clinical dose, and you see what is happening there. They are the cells. At the end, is disturbing the microtubules of the cell. It's like it's a pity that I didn't put the picture here. The microtubule, we take the cells, we stain the cells, and you have these 9 microtubules, these lines, these pretty pictures, for the papers. And when you put the colchicine, the microtubules is like all over the place. The cells stop dividing. And in immune cells, it's pretty nice, but because that is necessary to take care of the amount of colchicine that you provide. But at the end, the clinic has been tested that is safe. But in no, the clinical. No, you don't need to be scared. But when I was talking about the transference of these results, that, we are using colchicine, it's not water, be aware that you need to imagine that somebody wants suddenly to, I'm going to double the doses. No. Just be calm and
yeah, it's highly toxic in high doses. So the only thing that I would like to add is the one slide that you showed, the biomaterials interaction. So the yeah, this one. So I think I got lost here, but this again is the discussion of the small pieces of metal that will be released, or other compounds like titanium, and that interacts with toll-like receptors, particularly TLR4, and inflammation that it induces. This is the stuff that
we did different approaches here. For example, we changed it. Well, we tested with hydroxyapatite. These do not work properly. What it works like this, and it's the cell. You have this specific roughness, not too much, not too low, and then we stimulate the cells with LPS or endotoxemia, and if we put the cells in this bed, they don't want to become so inflammatory. And if we put these cells in this bed, they do not choose this fate. They choose this because the roughness changed how the cytoskeleton of the cell is attached. Therefore, it changed the shape. And if you change the shape, you cannot change your body to accumulate fat. Additionally, we pick the ones that were more nice. We can control this. And we can control this. Therefore, altogether, is a nice method to develop implants that cannot be patented.
Okay, so you're explaining to us that the laser treated, meaning you're doing to the titanium plates,
yeah,
this kind of ruffle border.
Yeah.
Okay. And with that, with the ruffle border, you get much less, how do you say this, bone resorption associated with the prosthesis, with implants.
Yeah, yeah, we are quite happy because, you know, at the end it's something that can be industrially scalable because you can have an implant. In fact, we apply for a European project and we almost have it. Because you can scale it in an industrial level. You have the implant and you have a laser, which is cheap. You do this, it's like maybe you need, I don't know, 60 seconds or maybe 1 minute for every single implant. You change the shape and it became, instead of brilliant, it's like mate, I don't know how to say it, mate. And yeah, but you touch and you don't feel it because it's nanometric. Yeah. And it was pretty nice.
Hello. Rodolfo, thank you for the talk and for all the enthusiasm. We are collaborating with On the Target through Patricia, mostly in the spondyloarthritis field. And I was looking also at one of your slides and thinking, especially in psoriatic arthritis patients, in whom we have increased permeability in the intestine and we have obesity also, but we also have lean patients that have metabolic syndrome but do not have obesity.
Yeah.
So thinking, for example, at IL-17, that is a major cytokine in this disease, do you think that increased levels of IL-17 can be the cause of increased permeability in the intestine for these patients, and we are seeing the outcome and not the cause for the problem?
It could be, but with patients like this that are in theory lean, it was a paper, I don't know, you know, I'm quite old, but it was a paper about the proportion of lean masa grasa, masa magra. Yeah, and it seems that they are lean, but when you go to the, and then you go to the lipokines levels, and these these patients have massive levels of leptin. Therefore, your question is if you think that we are going to see something related with the gut dysbiosis in this patient. I guess yes. because at the end, you have a class, it's a psoriatic arthritis is quite related with, with this metabolic environmental disruption. I think you can try with a small cluster of patients, just picking a few these things.
And I had a question also regarding the leptin and adiponectin. So as you know, we are doing the TASTY study. We have finished the TASTY study, and now we are looking at the results. And so these are patients with rheumatoid arthritis, and half of them didn't have an intervention, and the other half had a diet with nutritionists, and they had the foods were being delivered at home. And now looking at the data, the patients that have the intervention, when we look at their feces, and the whole feces were like liquefied, and they were simulating cells, cell line that was from the gut. And what happened was the feces after the intervention simulated the cells releasing less interleukin-8 than at the beginning. So when we look at the raw data, each one of us, our feces are going to stimulate interleukin-8 in a different way. So some people would have 4,000, the other ones would have 100. So it's really different. But when we look at intervention, the great majority is going half. So it's really decreasing, the difference is statistically significant. So in terms of the feces after the intervention, after 12 weeks of Mediterranean food with fermented food, it really makes a difference in terms of what is releasing interleukin-8. So my question is, should we go and look at leptin, adiponectin, and all the other cytokines, because the patients are losing weight? That's something that is there.
Therefore, you are going to see these changes, because leptin is directly produced by the amount of fat.
Yeah, and
if you have 5 kilos of fat, you are going to have 5 units of leptin. You have 3, you're going to see a downregulation.
So you are doing our proteomics. So if your proteomics doesn't catch leptin or adiponectin because the concentration is really low, so probably should go into this and also do ELISAs or other techniques to make sure that the levels, because then we can make a connection between what's going on, and we are seeing in our patients that they are losing weight and they are losing the fat mass, and what's going on in terms of inflammation and endotoxemia.
Yeah. I think you are going to have benefits in both sides in the TASTY trial, because losing weight is losing, because that I also pick here all the information of my work in adipokines, because you lose weight, you improve, you reduce this inflammation. And also, due to the diet, you are going to provide polyphenols and these kind of things. Therefore, you are attacking in two places. I think it's worth it.
So we should do that.
I think you are going to show it, because it has been described hundreds of times. If you lose weight, your adipokine level go down.
We can connect it so it's a better story.
Yeah, for sure.
Yeah.
You have 2 ways to explain what you're seeing.
I think it's a good idea. Thank you. More questions from the audience? Yes.
Thank you so much for your talk. I wanted to go back to the amitriptyline and colchicine study, which results are very intriguing. I can imagine that in your population, men take much more colchicine than women, and I guess that women take much more amitriptyline than men. Did you perform a separate analysis comparing only men and women, and if so, did you get the same results?
Yeah, I don't have the slide here, but it's the paper is free. This is the paper. I think in the supplementary data is provided the sex separation of the analysis, and obviously the majority of the gut patients are male, because in the clinic it was amazing. This is really amazing. The amount of people in this cohort of patients in Santiago. This is the healthcare population, and our hospital is the area of the patient are more or less 500,000 patients, something like this. Even more if you take into account the consults. But these are only about 65, and when we split it in male and female, the behavior is similar. Nonetheless, the amount of patients in the females cohort is really small, because women are not so crazy in terms of the control, because for example, when you provide, it was a study from the Spanish Society of Rheumatology, and the adherence of the gut patients to the treatment is only 11%. 11%. You prescribe the drug, and only 11 out of 100 take properly the medication. And in women, they have less incidence because they control the diet, they do the things properly, and they have less of the disease. And also, they are more attached to the medication.
So you got the same negative correlations in men and women?
Yeah. Similar findings were observed after sex stratified analysis. It is in the super. Yeah, I know this is a limitation because we cannot access to this data. Because otherwise, I would sign papers till the end of the days. Because having this information is there are quite a lot of patients. This is a tough question and a very good question. The reviewers of the British Journal of Pharmacology ask the same. And I say, well, I don't know, because it's difficult to transfer the information. I think it's within the therapeutical range of amitriptyline, in the top range of the amitriptyline dose, you could have some effects. But it's difficult to say because extrapolated concentration from the cultures is difficult. Taking this into account, it seems that if you take amitriptyline as this is drug bosses, and we calculate with the pharmacy of the hospital, and it seems that above within the therapeutical range in the upper part, you should start seeing something. But it's not, this is not a proper study. It's just, being smart in the hospital bureaucracy to have some data and to attach, to support our basic data. It's difficult. But if I need to bet, I would bet in the upper part of the therapeutical range.
Thank you.
Yeah, you're welcome. You need to be aware because for patients with cardiovascular alterations, amitriptyline sometimes is a little bit. Yeah, you can also, if you provide this at night, they're going to sleep like babies. Therefore,
So thank you so much, Rodolfo.
You're welcome.
It was wonderful. Thank you.
You're welcome.